and parasites. Long we'll notice that however certain diseases, for example, withers, are not attacked by the same disease again. Clearly, these people have become immune to the concerned disease. The system animal body, which protects it from various infectious agents and cancer, is called immune system. Immunology, the Latin immunis meaning exempt or freedom, gave rise to the English word immunity. It refers to all mechanisms used by body for protection from environmental agents that are foreign to the body. These agents may be microorganisms or their products. Certain food items, chemicals, drugs, and pollen grains immunity is up to types. Innate B. Acquired immunity. Innate immunity. Non-specific. Innate immunity comprises all those natural defense mechanisms with which an organism is protected from infection. As a strategy, innate immunity consists of various types of barriers that prevent entry of foreign agents into the body. The pathogens that enter into the body are quickly killed by some component of the immune system. This is the first line involving four types of barriers. Anatomicals. These barriers block entry of organisms into the body. The skin and the mucous membrane lining of the respiratory and intestinal as well as the reproductive passages constitute the barriers. Mucous material interrupts foreign microorganisms. The ciliary movements produced by epithelial lining cells expel out microorganisms from the body. Physiological barriers. Factors like body temperature, pH, and various body secretions prevent the growth of pathogenic microorganisms. For example, fever response inhibits growth of many pathogens. Acidity of the stomach contents due to HCl secretion kills ingested microorganisms. Lysozyme present in secretions such as tears and saliva digest bacterial cell walls. Certain white blood cells, when infected with a virus, respond by releasing antiviral proteins called interferons. Interferons in turn make the cells in the vicinity resistant to viral infections. As a result, the concerned persons exhibit increased resistance to viral infections. Phagocytic bacteria barriers. Phagocytic phagocytosis is an important mechanism of innate immunity. It is performed by leukocytes in response to pathogenic infections. The total count of leukocytes will increase sharply. Humans contain wandering phagocytes that circulate throughout the body. The most important phagocytes are macrophages and neutrophils. Macrophages are large, irregular shaped cells that engulf microbes, viruses, and cellular debris. In response to an infection, monocytes are liberated at the site of infection. These monocytes get converted into macrophages. Cells are provided with bacteriolytic enzymes and free radicals, which destroy the pathogens. Inflammatory barriers, usually an infection or tissue injury, results in redness and swelling, along with pain and production of heat that may result in fever. The above phenomenon is known as inflammatory response due to release of chemical alarm signals, notably histamine and prostaglandins by damaged mast cells. At the site of inflammation, there may be leakages of vascular fluid, which contains serum proteins with antibacterial activity. Further, there is an influx of phagocytic cells into the affected area. These responses inhibit and destroy the invading microorganisms. Besides the phagocytes, natural killer cells T lymphocytes kill virus-infected cells and some tumor cells of the body by creating perforin-lined pores in the plasma membrane of the target cells. These pores allow entry of water into the target cell, which then swells and bursts. Acquired immunity, specific immunity. Acquired immunity, also known as adaptive or specific immunity, is capable of recognizing and selecting. Acquired immunity is found only in vertebrates. It supplements the protection provided by innate natural immunity that is generated in response to an exposure or encounter to the microorganism in question. The mechanism requires several days to be activated. Following the mechanism, adaptive immunity has the following unique features. 1. Specificity. It is the ability to distinguish differences among various foreign molecules. 2. Diversity. It can recognize a vast variety of foreign molecules. 3. Discrimination between self and non-self. It is able to recognize and respond to molecules that are foreign, non-self to the body. At the same time, it can avoid response to those molecules that are present within the body. Self antigens of the given animal. Memory. When the immune system encounters a specific foreign agent, for example, a microbe, for the first time, it generates an immune response and eliminates the invader. The immune system retains the memory of this encounter for a prolonged interval. As a result, a second encounter with the same microbe evokes a heightened immune response. Specific immunity implies two major groups of cells, lymphocytes and B antigen presenting cells. A healthy individual possesses about a trillion of lymphocytes. Lymphocytes are of two types: T lymphocytes are T cells, are B cells. Both the types of lymphocytes as well as other cells of the immune response are produced in bone marrow. The process of their production is called hematopoiesis. Some immature lymphocytes tend to become thymocytes migrate to blood the thymus, where they mature and differentiate as T cells. The B cells, on the other hand, mature in bone marrow itself. The B and T cells together generate two types of specific immunity: cell mediated, antibody mediated, humoral immunity, respectively. Cell mediated immunity. Cell-mediated immunity is the responsibility of a subgroup of T cells called cytotoxic T lymphocytes. An activated cytotoxic lymphocyte is specific to a target cell which has been infected and kill the target cell by a variety of mechanisms. Thus prevents the completion of life cycle of the pathogen and its growth, since it depends on an intact host cell to do that. Cell-mediated immunity is also involved in the killing of cancer cells. Antibody-mediated immunity, humoral immunity. Antibody-mediated or humoral immunity involves the synthesis of specific antibody molecules called immunoglobulins by the B lymphocytes. Each antigen has many different antigen determinants, each of which matches a specific antibody and bind to it. The B cells direct the antibody mediated immunity. The antibody molecules may be bound to a cell membrane in the form of receptors or they may remain free. The pre-antibodies have three main functions: of particulate matter, including bacteria and viruses; 2. Opsonization or coating over bacteria to facilitate recognition and phagocytosis by phagocytes; 3. Neutralization of toxins released by bacteria. Adaptive immunity may be active or passive. Active immunity is due to immune response generated in individual in question by pathogen or vaccine, where a specific immunity is confirmed by transfer, like antibodies, for example, from an individual into a non-immune individual. Activation of adaptive immunity. Every antigen is processed by antigen-presenting cells like macrophages, B lymphocytes, and dendritic cells. The processed antigen is presented on the surface of these cells. A subgroup of T helper cells specifically and becomes activated. The activated T helper cells then activate beta cells, B cells, and a subgroup of T cells called cytotoxic lymphocytes. Lymphocytes proliferate to produce clones. All the cells of a clone structure and function of immune system. The lymphoid system consists of lymphoid cells and lymphoid organs based on different lymphoid organs can be classified into central, primary, and peripheral. Lymphoid organs, the central lymphoid organs are lymphoepithelial structures in which the precursor lymphocytes proliferate, develop, and acquire immunological capability. And mammals, the represent accumulate accumulate in peripheral lymphoid organs and following antigenic stimulus affect the appropriate immune response, the spleen, lymph node, and mucosa associated immune response, and mucosa associated lymphoid tissues. The major peripheral or secondary lymphoid organs. Lymphoidal tissues in the gut, appendix, tonsils, salivary gland, tear gland, and also the secretion of the lactating breast also are included in the immune system. Primary lymphoid organs, thymus. The thymus develops at about the sixth week of gestation. By 8th week, it grows into a compact epithelial structure. Mesenchymal stem cells, precursors of lymphocytes from the yolk sac, fetal liver, and bone marrow reach the thymus and differentiate into the thymic lymphoid cells. Thymocytes. Its characteristics lymphoid appearance by third month of gestation, and thus the first organ in all animal species to become predominantly lymphoid. In human beings, the thymus reaches its maximal size just prior to birth. Thymus continues to grow till about the 12th year after puberty. It undergoes, it undergoes spontaneous progression, indicating that it functions best only in early life. The thymus is located just behind the upper part of the heart. It is two lobes surrounded by a fibrous capsule. Septa arising from the capsule divide the gland into lobules, which are the cortex, and an inner medulla. The cortex contains small lymphocytes. The medulla consists mainly of epithelial cells and mature lymphocytes, which are the hostus corpuscles or wall-like aggregation of epithelial cells. The thymus was considered as an organ without any recognized function, but its role in the development of cell-mediated immunity has been found recently. The primary function of the thymus is the production of thymic lymphocyte T cells. It is a major site of T lymphocyte proliferation in the body. However, lymphocyte produced only about 1% leave the thymus. The rest are destroyed locally by programmed cell death or apoptosis. In the thymus, the lymphocytes acquire antigens. Lymphocytes produced in the thymus are called thymus-dependent lymphocytes. At proliferation, In the peripheral organs, the function of thymus is independent of antigenic stimulation. The thymus confers immunological competence on the lymphocyte during their stay in the organ. Thymic lymphocytes are not immunocompetent. In the thymus, they are educated so that they become capable of mounting cell-mediated immune response against appropriate antigens. Thus affected by hormone-like factors produced by the thymus such as thymoline, thymosin, and thymopoietin. Thymus in lymphocyte proliferation and development as evident from the effect of lymphopenia and in neonatally thymatomized mice. Spleen. It is the largest lymphoid organ. It contains red and white pulp regions that serve as filters. The macrophages within the spleen help to remove and destroy pathogens. Function of spleen. The spleen serves as the graveyard for aged red blood cells. It acts as reservoir tank and sitting right for blood, and it acts as a systemic filter for trapping circulating blood-borne foreign particles. The immunological function of the spleen is primarily directed against blood-borne antigens. Secondary lymphoid organs. Antigens. The term immunogen and antigen are often used synonymously. However, these terms simply to closely related entities. The first describes a molecule that provokes an immune response and hence is called immunogen. The other describes a molecule which reacts with the antibody produced or with the activated cellular constituents of cell-mediated immunity and is referred to as an antigen. In contrast to this is the heptane. Heptanes are small, well-defined chemical groups such as dinitrophenol, which are immunogenic on their own but will react with formed antibodies to make a heptane immunogenic. It must be linked to carrier molecule, which is itself immunogenic. Antigens are recognized not only by antibodies but also by antigen-specific T cell receptors in contrast to immunoglobulins, which usually recognize intact antigens. T cells recognize processed antigens on the surface of antigen-presenting cells. Antigenic determinants and epitopes. The part of antibody molecules which make contact with the antigen is termed as paratope. Consequently, the part of the antigen molecule that make contact with the paratope is called the epitope. As most antigens are protein in nature, they exist in a folder, three-dimensional tertiary structure. Hence, there may be a closure of amino acid dimensional structure constituting a series of epitopes. These epitope clusters on the antigen are known as an antigenic determinant. Generally, the substances which are capable of eliciting an immune response that is synthesis of antibodies are called antigens. However, the above definition of antigens do not infer the cell-mediated response. Hence, the term immunogen is introduced to denote antigenic substances which elicit both humoral and cell-mediated immune response or either humoral or cell-mediated response. Antigens may be either natural or artificial synthetic ones. Natural antigens fall into two categories: particulate antigens and soluble antigens. Lymphocytes and self represent particulate type toxins, proteins, carbohydrates. glycoproteins, and lipoproteins represent the soluble antigens. In addition, a variety of chemical compounds, biological macromolecules, synthetic polypeptides are regarded as potential antigens. Several polysaccharides, nucleoproteins, and lipoproteins are also considered as antigens. Recent antibodies to DNA have been used in immunization. Antibodies. Immunoglobulins. Immunoglobulins are glycoproteins. Each molecule of ID consists of two pairs of polypeptide chains of different sizes. The smaller chains are called light L chains and the larger called heavy weight of approximately 25,000. H chain is a 50,000. The L chain is attached to the H chain by a disulfide bond. Depending on the class of immunoglobulins, the H chains are structurally and antigenically distinct for each class and are designated by the Greek letter corresponding to the immunoglobulin class as follows: IgG gamma, IgA alpha, IgM N, IgD delta, and IgE epsilon. The alkanes are similar in all classes of immunoglobulins. They occur in two varieties, kappa and lambda. A molecule of immunoglobulin may have either kappa or lambda chains, but never both. Region of polypeptide chains. Each heavy and light chain consists of two regions. Transplantation immunology. The replacement of a diseased organ by a transplant healthy tissue or organ is called transplantation. In the early 1940, Medauer explained the nature of graft transplant rejection while working with the burn patients of World War II. In his observation, the following features were made clear. Graft of skin from one region of the body to another in the same patient was easily accepted. Grafts obtained from close relatives like brother or sister were rejected. When a second graft was performed by obtaining the tissue from the same donor, the rejection reaction occurred with greater intensity and speed. The graft or transplant lead to various complications in the host body. They are mediated by the host immune response. Very often, the transplant gets rejected or may lead to graft versus host reaction or diseases. Before discussing the nature and implications of this rejection phenomenon, let us look into terms involved in various types of grafts. Classification of graft. The graft can be classified into four major types. For example, skin graft from thigh to face in severely deformed case of burnt individual plastic surgery. 2. Isograft. Graft between syngenic individuals, identical genetic constitutions, for example, clones or identical twins. Allograft. Graft between allogenic individuals, members of the same species but different genetic constitution. For example, kidney transplanted from the one human to another. Xenograft. Graft between xenogenic individuals, different genetic lineage, for example, organ transplanted from pig to humans, baboon to human, etc. Genetic basis of organ transplants. Immune system disorders. The immune system is a multi-component interactive system. It effectively protects the host from various infections, but an improper functioning of the immune system can cause discomfort, disease, or improper function of fall into the following major groups: hypersensitivity or allergy, autoimmune disease, and immunodeficiency. Hypersensitivity. Allergies result from inappropriate and excessive immune response to common antigen substances that cause allergies are called allergens. They include dust, molds, pollen, certain foods, and some medicines such as penicillin. Allergy involves mainly IgE antibodies and histamine. Most cells secrete histamine, a common manifestation of allergy as asthma. Sometimes an allergen may cause a sudden, violent, and fatal reaction in a sensitive individual. This is called anaphylaxis. Autoimmune diseases. Autoimmune diseases is when the immune system attacks and destroys cells and molecules. This condition can cause chronic and serious disease. Example of autoimmune disease are insulin-dependent diabetes, multiple sclerosis, rheumatoid arthritis. Multiple sclerosis is caused by antibodies that attack the myelin sheath of nerve cells. Immune deficiency disease. Immune deficiency disease results from a defect in one or more components of the innate or adaptive immunity. Affected individuals are susceptible to disease that normally would not bother most people. Immunodeficiency may result from gene mutation, infection, malnutrition, or accident. Severe combined immunodeficiency results from one of many genetic defects. One such genetic defect leads to adenosine deaminase deficiency. SCID is characterized by a very low number of circulating thymocytes. Affected individuals usually die at an early age. AIDS, acquired immunodeficiency syndrome, as another example for immunodeficiency disease. It is caused by retrovirus, known as human immunodeficiency virus. Retrovirus has RNA genomes that are replicated via DNA copies. HIV selectively infects and kills T helper cells. The depletion of T helper cells weakens the acquired immune response and may even abolish it completely. The viral RNA genome is converted into DNA copy by the viral enzyme reverse transcriptase. The DNA copy of HIV becomes inserted into the human chromosome and replicates with the cell DNA. It may be transcribed to produce RNA copies of the viral genome. The RNA copies are packaged and labelated as virus particles. The infected cell is lysed in this process and the released virus particles infect new T helper cells.